Hello all, and thanks for the invitation today. I'll start off with a couple of definitions. I saw you have a glossary here of terms, and proteomics is one of those. So we know what proteins are, it tells us here. Large, complex molecules that play many critical roles in the body. They are responsible for most of the functions in the body's cells and are required for the structure, function, and regulation of the body's tissues and organs. Nicely done, Fernando. It's good definition. But then there's this term proteomics, and people may have heard the term genomics. So we know what genes are. There are risk genes for ALS, such as SOD1, superoxide dismutase. But the study of all of them in concert and the holistic view of all of the genes we have in our body is genomics. And so by proxy, then the study of the mixture of different proteins that we have in our cells and in our bodies is proteomics. The scientific field has gotten a little bit carried away with the suffix. It's a little bit like Snoop Dogg, whom you may have been reminded of in the Olympics this summer with his use of "isl" at the end of every word. But the scientists have gotten very excited, so you'll hear metabolomics, you'll hear epigenomics. In the end, it's a systems study of what cells are and what is happening at the biological level. And it's really remarkable in our progress to be able to have gone from measuring a single thing when I was in the lab first learning how to do the pipetting and the aliquoting that Fernando talked about, to now really being able to, in parallel, measure thousands of different things in that same amount of time. So we're at a revolution of data that, as Danielle pointed out, a lot of conventionally trained scientists are drowning in, and yet the potential for us to make these insights and make these next discoveries is sitting in front of us, but we don't necessarily have the tools always to bring that data together or to make the most of it. So I'm going to tell you about two things today. I'm going to tell you about this proteomics project that not only generates new data, but also has brought together a global set of data that existed but was separated and siloed into what has become the largest discovery data set for any disease in history. And it started off, I'll tell you the story about it. It started off during COVID with people scratching their heads saying, gosh, what do we do now that we can't get two people in the same room to actually do a clinical trial anymore? And the second part is a software platform that helps do a lot of what Danielle was telling you about, which is how do we put the tools to find those needles in the haystacks in the hands of as many doctors and scientists as possible without requiring them to retrain as bioinformaticians and computer scientists, and without requiring that they learn R and Python and MATLAB and Java and the different computer languages. And so those are the two things that I'll tell you about today. The last definition on here I'll tell you about is Gates Ventures. Sounds like a strange organization to be involved in research in disease, but my own background comes from having been a physician scientist in the university, having done a sabbatical at the Bill and Melinda Gates Foundation in global health, and having worked on biomarker discovery, drug development, and devices for global health disease across HIV, tuberculosis, malaria. But then when we wanted to work on non-communicable diseases, that is to say diseases that are not spread by viruses or bacteria, that is not yet part of the World Health Organization's strategy. And so we could not work on it at the foundation. And so starting with a personal experience, as both Danielle and Fernando, and many of you in this room have, disease is very personal. And so the movement to work on Alzheimer's started because of Bill Sr. and a desire from Bill Gates to work on that disease, but we could not within the structure of the foundation. And so we moved the work. Again, a big philanthropy approach, working with doctors and scientists around the world, but that same nonprofit philanthropy style into a separate vehicle that we were able to execute on. And so that's why we were working on neurodegenerative diseases like Alzheimer's, like Parkinson's, like ALS, like frontotemporal dementia, in a very similar way, but just in an adjacent org. So a little bit of background for those pieces. Coming back to the origin story of the Global Neurodegeneration Proteomics Consortium. Gosh, it's a mouthful, but we'll call it the Proteomics Consortium for short. I mentioned that when COVID hit, a lot of research got hit very hard. So clinical trials were no longer able to run in the normal way they could, because it's hard to be 6 feet apart and masked and feel safe from an infection point of view. And as Danielle was saying, it was an extra step for so many families, an extra step for health systems, that this really fell apart for a period of time. And so then the question became, how do we still move the field forward? How do we still make progress on diagnostics and treatments in the setting of a pandemic? And there was a conversation between our organization and a pharmaceutical company, Johnson & Johnson, and we're part of many public-private partnerships. Some are brokered by the NIH and their foundation for the NIH program. Others are independently set up. And the thought was, well, we have all of these samples in freezers because of participants in studies, like some of the people in this room who over the years have donated biosamples and data to building repositories. ALS TDI has an excellent example of one of those, and they're actually part of this consortium, and I'll come to that in a minute. But and then the thought was, well, we often run out of budget because we have the clinical trials to run. This is what we're hearing from the pharmaceutical companies. But now we can finally focus on really the fingerprinting, coming back to the detective work, the biological fingerprinting of these samples that have been sitting in freezers that we've only scratched the surface of in the meantime. And so we said to them, well, you know, if you're going to do that, if you promise to put that data out in the public domain so that everybody gets access to it, we will match the number of samples that you generate data from. And so initially we thought, well, maybe we will each do several thousand samples and put together something like 5 to 10 samples in aggregate. Well, this ended up becoming the largest proteomics discovery data set to date, and includes 40,000, roughly 40,000 proteomic assays from blood samples. And so I'll tell you a little bit about the data that's coming from it in a minute. But one of the things about the Proteomics Consortium is that it's intended to grow annually. So if you see that little symbol on the bottom left, it's a Mobius strip. And so the thought is that annually we will continue to bring more and more data in. We will continue to bring more and more scientists and clinicians into the same environment. And it's not just the value of the samples in the end, as we look back, that's been created here, but it's also all of the scientific expertise that's coming together in the room and thinking, gosh, what more could we do? How much more data could we share? And it's really been a beautiful experience to see that conversation happen. Whereas traditionally data, as you've heard from Danielle, is often separated and hard to connect and bring together. So 4 key diseases here. In addition to ALS, there's Alzheimer's, Parkinson's, and frontotemporal dementia that are predominantly represented. And I'm going to show you what I think is a good analogy for the big data, the type of big data that Danielle mentioned earlier. So if you think of a single protein that you're measuring and a static time point, that's just one pixel that you're looking at. But if you really want to understand the trajectory of a patient in disease, or the trajectory of hundreds or thousands of patients across disease, you need the kind of longitudinal samples over time, or at least a sample with longitudinal follow-up that tells you what happened and what kind of trajectory a particular patient or participant had. And so this starling murmuration is something that I think is a good example, and you'll have to tell me if a click is going to be enough, and it looks like it is. So what you're seeing here is actually thousands of individual trajectories of birds in flight that are being traced in 4 dimensions. You're adding the dimension of time to this. And there's an amazing artist from Barcelona that does these. And as soon as I saw these, I thought, gosh, this is how we can now visualize patient trajectories in a way that we never could before, because we have thousands of data points for each and we can look at them all in aggregate. But really, what we wanted to do is look back at, this is where we used to be in thinking. We didn't know where a particular patient would go. We didn't know how somebody's disease was different from somebody else's disease. Is there a chance that one drug or treatment would work for one patient that would not work for another patient? So this gets to subtyping. Some of the NFL work that uses one protein to try and do that. Now we can look at 7,000 proteins and really do a fine fingerprint and say, okay, here are different subclasses of disease. We already know, and in fact, ALS is one of the leaders in this disease space of saying, if you have this particular mutation, if you have this particular disease type, this drug will work for you. And this is a better method of management for you. In some ways, we aren't there yet in Alzheimer's, and ALS has the advantage of being there also with its lead in innovation in personalized therapies with siRNA. So we have a lot to learn from each other in these adjacent disease spaces. And by pooling all of our data together, we really learn what the common threads are from these neurodegenerative diseases, in addition to how they separate and how they're different from each other. And that's really, if you look at cancer as a field, the progress that they have been able to make with multiple therapies, that are optimized for a particular type and subtype of patient and disease, and how it's dramatically changed outcomes and trajectories for patients there. That is where we should be able to get, and indeed we're on track for getting, by combining technologies and data like this with the participation of the people like you in this room, as well as the organizations like ALS TDI. So this is the goal, right? The goal is to identify in a personalized way what is the trajectory of each of these individual patients and what is the best combination of therapies that we can bring to bear. We have some today, and so we need to increase the arsenal. We need to increase the number of tools we have available, but we also need to know who exactly is going to benefit from it in which way. So when we started to set this consortium together, we thought, well, let's bring together some of the best researchers and data that's out there. Let's accelerate this upstream discovery space. Let's leverage the strength of the cloud. That's actually something that came out of the pandemic, right? The hybrid work environment, our ability to work with each other at a distance, but also the ability to share data together across long distances using the secure cloud. How can we harmonize these kinds of data from different groups? You heard about OMOP as one of the methods, having this common data model, common data system, so that underneath what each different center is doing, we can find those same apples to apples comparisons of the data and be able to combine it into one larger data set. And then we wanted it to be low lift and relatively straightforward so that we could attract other data sets. And so here's where we are today. We have over 30 different cohorts that we are closely engaged with. 24 of them are actually already in the V1 of the data set. And this is data that is already in the hands of the people of these different groups that are in the consortium, including ALS TDI and Fernando's. And 1 year after that data is analyzed by that internal team and quality controlled, it goes out to the entire world. And this is an annual cycle that we're initiating, and so we're already starting with trying to recruit V2. And it started with Gates Ventures and Johnson & Johnson, but we're expanding now to other pharma, other philanthropy who are interested in joining and accelerating this creation and sharing of data for the good of the global research community. Another thing we did is we brought a professional data harmonization team in. And so having been a professor in academia before moving to the foundation and ultimately into the ecosystem of big philanthropy, a lot of this work was on the shoulders of postdocs and grad students, and pharma had the benefit because they had the budget of doing this for clinical trials. But what we said was, we're going to do this fast, and we're going to do this at a level of professional quality that you don't generally get in the public sector. And because of that, we were able to get the buy-in of thousands of other data points and multiple other cohorts that already had their data sitting there and said, wait a minute, you're going to do the hard work of harmonizing this data that we've already generated with all this other new data that you're doing. Here you go. This is great. This is exactly what needs to be done. But in general, it's a big hurdle, and it's hard to overcome that piece because nobody tends to fund that. Again, this is the role of philanthropy, is to find, big philanthropy from our perspective is to find the gaps between what industry does and where they go, what the public sector and NIH does and where it goes. And so we don't want to be redundant with either of those two groups. We want to find the gaps that are the most important for researchers, patients, families, and try and move those and address those as quickly as possible. So you'll see here that there's 4 groups in the ALS space. In addition to ALS TDI, there's Answer ALS, Target ALS, and the University of Edinburgh has an interventional 4-arm trial in ALS that was originally going to be part of V1, but there was enough concern about potential unblinding while that trial was happening that we're holding off until that trial completes, and then we're bringing them in to V2 of the consortium. Target ALS will also be in V2 of the consortium. So at a glance, what have we ended up with? 24 cohorts, about 40,000 analyses, 300 million independent protein measurements. So I want to stop there for a second, because when I was a graduate student in biochemistry, it would take me a week to do what's called a Western blot and measure 1 protein. And so this number is staggering. So you know, granted that was a few decades ago, but for us to be able to go to measuring thousands of proteins in the same amount of time that it took to measure 1, and then to bring all these data together, this is really an unprecedented opportunity and challenge that we're sitting on right now, which is, we're not looking at bands on a gel. We're not looking at data with our own eyes anymore. You heard a little bit about how can we leverage artificial intelligence? How can we leverage computation to take us to the next level? And I have a little example of that as well coming. But let's focus on the data sharing piece. So this sounds great. You'll see the word legal on there. Okay, I'm glad that we already talked about detectives and law and order. So law and order generally says, gosh, sharing data is risky and we're in different jurisdictions and it can't be done. And so when we started to do this, we were told that it was impossible. I recently read Steve Gleason's book, A Life Impossible. And the Team Gleason signature of no white flags, right? So we're not going to stop when people tell us that things are impossible. And you know, that extremely challenging and also extremely beautiful story that he told, that is the story of many people in this room as well. We are not going to stop because people tell us something is hard or impossible. And so what we were able to do for the first time is to bring together data that was regulated differently in the United States than how it's regulated in Europe. And the UK, which used to be part of Europe but isn't anymore, is a third difficult stepchild. And so how do you get all these people to agree? Even in the US, we have so-called covered and non-covered entities. And so after about a year of going to half a dozen different dementia centers and being told no, 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 there was a point at which we almost gave up on this project. But having come from a background of being a discovery scientist and failure being the norm and innovation being what you get because of persistence, sometimes a good thing, sometimes a bad thing. In this case, a good thing. We ended up finding language, common language, that 6 or 7 top centers were excited and willing to sign. And so once we had the NIH, and we had WashU St. Louis, and we had Banner Alzheimer's Institute, and we had Emory University, and we had UCSF, and we had Stanford, then the next person coming to the table could say, well, I don't like the language in paragraph 3, section 5. And I would say, tell me again, what about that you don't like that the NIH, that UCSF, that Stanford, that Harvard were fine with? And so then it flipped. There was this really interesting tipping point where people started coming to us and saying, gosh, we've heard about this really exciting thing that you're doing, and you figured out how to get all the data into one place. Can we be part of that too? And I kind of felt like I had woken up in a different universe, because for a year I had been told, no, this is impossible, it'll never happen. And then very quickly it transitioned to, this sounds great, can we do it too? But I think it's a good lesson in finding the right solution that ends up being acceptable to multiple different parties. And I'm sure that many of you have been part of things like this, but this tipping point was really fascinating for us to be able to experience. And now we can do this, right? So now we have that data set, and there are over a dozen different research groups in core work streams that are actively analyzing this and trying to get all of the different insights that we can get from this data set. So some of this is what's common across all of these diseases and what's a biomarker of neuronal injury in general. NFL falls into that category. But what about disease-specific markers? What about signatures that are more than a single protein? So all of these things now become possible, and I'll give you a small taste of what that looks like in the preliminary analyses. But we're already looking at V2 because that cycle is now coming for the second year. And what you see here is that there's over a dozen cohorts that we've spoken to and are excited to join, but are not yet part of V1 and have not yet had the analyses done. And so we're bringing together a coalition of funders, both public and private, to put resources together to generate these new data from these additional cohorts. You can see a couple of them are in the ALS space, both Target as well as Edinburgh, that has this 4-arm, 4-intervention study. But thousands of additional samples here that'll add complexity, diversity from both a geographic as well as an ethnic perspective, and further kinds of disease variants. So here's one graph that was shown in the very first data report out from when people had these harmonized data set in their hands. And what somebody showed that was with about 100 protein signatures, so that's a few % of all of the proteins that are measured, 7,000 in a single drop of blood, they were able to say they were able to put with a very high level of prediction what degree of cognitive impairment in the Alzheimer's disease space people had. So you could predict the quintile or the fraction of cognitive impairment that somebody was in without looking at a medical record, just looking at a specific sub-fingerprint of about 100 proteins. The other thing that people were able to do with this data set was to say, gosh, we've found this signature that tells us ALS versus Alzheimer's very earlier in disease, or we found this signature that tells us rapid progression of disease versus likely stability, prognostic biomarkers. These are signatures that individual groups found by looking at 500 or 1,000 of their own samples. They could have done that on their own. What they were able to do here is with the push of a button, immediately validate that exact same signature across 40,000 other samples. And so the acceleration of the ability to validate something that's a preliminary result is also something that we've seen come out of this data immediately. And in the final couple minutes, I'll tell you about this global research and imaging platform called GRIP. You might ask, gosh, what does imaging have to do with ALS? And the answer is, well, this started from fetal ultrasound and infant MR needs in that research space, as well as Alzheimer's disease, where researchers were having difficulty sharing data across with each other. And this was back actually started when I was at the Gates Foundation, where we were literally flying hard drives of fetal MR ultrasounds between Kenya and Pakistan, because that was the best way to share that imaging data. And so the need for a secure cloud environment and the ability to cross-annotate and share data, then that quickly expanded to be more than just imaging data, but also omics data, also digital data such as speech. And ultimately, having built a couple of these pieces, we recognized that putting it together into a platform that would be free for the public community and the research community could be of value. And so we've partnered with leading dementia research centers to build some of the core components of this, which were their initial pain points, but then putting them together, beads on a string, into a coherent platform where you can do a start to finish workflow, going from what comes off of a scanner, off of a sequencer, all the way through to cleaned, quality controlled, prepped data that you could then send to build a machine learning model or to do conventional analytics. And I'll just finish with something that we've done here. It's a beta version, but it's a large language model, or basically a chatbot for those scientists and doctors that may not be fluent in programming at the command line. And what you're seeing here, and it's probably too small to see, is they're just typing questions of the data set. For instance, how many patients are in this study? And you're seeing the code that it's writing. This is actually in real time. This is the real thing that it's doing that I took a snapshot of. How many patients are in this study? What's the gender distribution? What's the age distribution? How many people have ApoE status, and what's the different sort in this Alzheimer's study? How does that correlate with their cognitive state? And then finally, can you plot that in a graph for me so that I can see the distribution in these different categories? And so again, this is a very early beta version of this that we've put out with a clinical trial that was a big readout trial called the A4 study, or anti-amyloid therapy for asymptomatic Alzheimer's. But it's just an example of the kinds of tools that we have the ability to bring now to these diseases and to the study of these diseases that we're interested in trying to facilitate, and the kinds of things that'll help us get our heads around these bigger and bigger piles of data that we're generating, but that are harder and harder for 2 human eyes and 1 human brain to be able to take in as a gist. So thank you for your attention. I'd be happy to take any questions.
Hi, I'm Mehreen. I'm asking this on behalf of Bernie. So question, can you comment on the All ALS initiative from the NIH? You've built such a wonderful foundation. Are efforts happening to ensure that the All ALS initiative builds on what you've created? And if not, how might we make that happen?
Yeah, so we are very interested in collaborating and working with and giving any of these tools to anybody that could find it useful. We have a long history of working with the NIH, both from the Bill and Melinda Gates Foundation, where we have an annual meeting from our discovery team, which I used to be on, and all of their directors of the different institutes, where we compare notes and say, gosh, what are the directions we need to go? I sometimes make the analogy of, if the NIH is the cruise ship, we sometimes have the option to be the speedboat. So we're much smaller than they are, but we can move together with them in directions that we share interest and insights in, and we can sometimes fund smaller things faster and easier than they can, but ultimately we link up in providing resources in both the short, medium, and long term to areas of high priority. And so that's a very similar way that we work with the Foundation for the NIH, which actually is an adjacent org that can bring together public-private collaborations like these accelerated medicine partnerships or AMP programs. And so both at NIA and different institutes within the NIH, we have active conversations and active collaborations. For instance, AMP AD for Alzheimer's disease 2.0. We provided both resources, but also are offering in-kind computational resources for data use across those different programs.
I noticed on the one slide it mentioned the effects of comorbidities, and I was wondering, are you looking at individual ones or the presence of comorbidities as the variable itself?
Yeah, it's such an important question as we talk about and think about personalized therapy and how different subgroups that are called one disease, everybody used to be called ALS or Alzheimer's or Parkinson's. We know in reality there are multiple different subtypes within those diseases, and comorbidities are one of the key pieces, not only that tell us something mechanistically about how maybe there are contributing factors to the mechanism and to the onset of disease, but also give us a clue as to, is this disease that looks very similar in these two individuals actually different, right? Did it arise because of inflammation rather than a genetic predisposition? Did it arise because of an auto-inflammatory or self-reactive process rather than something that is inherent in the neurons in the tissues themselves? And so comorbidities are incredibly important to capture both clinically in the record, but also for us to understand mechanistically. And so for instance, with type 2 diabetes and obesity being, for instance, a comorbidity that puts patients at higher risk for certain types of neurodegenerative disease, we are right now taking a learning perspective and learning more about that other non-communicable disease and its mechanisms to think about how best to also include that potentially in the portfolio of research and things that we're pursuing because of exactly what you say. So it's not only important to understand the disease itself, but also all the associated other comorbidities that are related to it. Thank you.